Okay, welcome to a video lecture on genetics for non-majors biology. Woo! So let's just get right into it with some vocab, right? So you need to know some new terminology. Genes, right? So a gene is, for our purposes, encoding a trait, right? So your gene is for a given trait. When we're talking about this, gene is actually for single protein, but so when we talk about genes, they code for a category, like eye color. And we talk about, oh yeah, my favorite category other. When we talk about a trait, it's governed by a specific allele within a gene. An allele is a genetic unit. So we'll be learning about Gregor Mendel, the father of modern genetics, and he discovered genetics before really discovering anything like genes or DNA. So he used the term allele, and it's just a genetic unit, and it's what codes for a specific trait. So if the category in the gene is eye color, then the allele might be for something like hazel eyes or other. So, genes and alleles are inherited from, anyone want to take a guess? You? Huh? Over there? That's right, mom and dad. Mom and dad are the people from whom your genes and alleles are inherited from. And each one gives you one allele. Now, this is important because everyone has a pair of alleles for each gene. So if you have your eyes right here, you might have one allele for hazel and one allele for blue eyes, and one came from one parent, right, mom, and one might have come from the other parent, like dad. Ooh, that looks really good. Anyway, so, and then mom might have or mom had herself two alleles, like maybe an allele for hazel and an allele for brown eyes, right? And she could only pass one of those alleles on. So you got the allele from mom for hazel eyes, but not mom's brown eye allele, right? Makes sense. So you get two alleles for every single trait, two genes essentially for each trait, or yeah, and bam. So we'll be talking about what genes you express or what alleles you express. Uh, Basically, this is when we say expression, right? It is literally what is showing what is being produced, right? Since you have two alleles, you might not be making the product of both of those alleles. So, for kitties, mom might have short fur and dad might have long fur, right? And what is expressed in the offspring of these cats. So they could have short fur, they could have long hair fur, but they won't be expressing both. They'll be expressing one. Like if I'm smiling, I'm expressing happiness, even though my student loan situation might leave me dead on the inside. So I'm expressing one of those things. Right, so gene expression is when you make something. It's the visible product of the alleles. And for our purposes, we'll be talking about traditional inheritance, as Mendel discovered, and that includes the concept of allele dominance, right? For every allele, you could have either a dominant allele or a recessive allele, right? So for every trait, or category, let's say like for eye color, right? You could have alleles for, let's just make up the alleles, blue, hazel, easy to write with, wow, hazel or brown, right? Because those are three eye colors. Now what we're just going to do is ignore hazel and pretend it doesn't exist because eye color is more complicated. And you know what? This example's getting out of the way. Screw you, example. It's going to be earwax. Yay! I'm going to be going into this anyway, but you might have wet, sticky, yellow earwax, or you might have dry, crumbly, white earwax. Okay? And one of these will be dominant over the other one. When we talk about dominance, an allele can either be dominant or it can be recessive. Wow, this is not okay. It can be dominant or recessive. A dominant allele is always expressed. A dominant allele is always expressed. If a dominant allele is present, the dominant trait, the trait it codes for, is expressed. A recessive allele is only expressed if both are recessive. Both alleles, remember you have two alleles for everything, both alleles must be recessive in order for a recessive allele to be expressed. Right? So in the case of earwax, wet earwax is dominant and dry earwax is recessive. So dominant alleles always expressed, recessive alleles expressed only if both alleles are recessive. So I like to use cats because they're adorable, right? So fur length in cats. Cats come with either short fur or long fur, and this is pretty simple genetics governing fur length. So you have alleles for long fur and short fur. We will often use a single letter to abbreviate for alleles, where a capital letter, wow, is dominant, and a lowercase letter is recessive. So a capital letter will be dominant and a lowercase letter will be recessive. We often try to use a letter associated with or easily mentally connected to the trait we're talking about, the category we're talking about. Since it's fur length, my example here is using L's. L's are also nice because the capital and the lowercase are very different, right? So you kind of want to watch out for difference if you're thinking of your own letters to abbreviate with. So the dominant allele is for short fur and the recessive allele is for long fur. So if you have two dominant alleles, it makes sense that you would express short fur, right? You got two dominant alleles, then the only information you have is for the dominant trait, so you will express the dominant trait. Same if you have two recessive alleles, the only genetic information you have is for the recessive trait, so you express the recessive trait. But what happens if you have a dominant allele and a recessive allele? Right? So now you have genetic information for either trait. So which one do you express? And the answer is all about that dominance, right? Remember, a dominant allele is always expressed. A dominant allele is always expressed. When present, you express it. That's easy. So, a recessive allele is only expressed when both are recessive. It's the only way you can get the recessive trait to be expressed, if both alleles are expressed. So there we go. Short, when it's two dominant alleles, when it's a dominant and a recessive allele, it's still the short fur, and it's only when you have two recessive alleles for fur length that you get that long fur. So there is a cat that is showing two recessive alleles. And here is a kitty, which is pretty cute. So this cat, we know, two recessive alleles. But this cat right here is adorable little kitten. What are its alleles? Right? A lot of people new to genetics will just assume that they have two dominant alleles, because that's how you get short fur. But remember, all you need is one dominant allele. So we don't know the alleles for this cute little kitty, right? This adorable little kitty, so fuzzy. Okay, anyway, it could be two dominant alleles, or it could be a dominant and a recessive allele. There's just no way to know without actually doing some genetic testing. All right, so in humans, you remember I might have mentioned this, right? There's only a couple of traits that are simple dominance recessive, like literally just a few. And earwax is the easiest one to know them. Cerumen, by the way, if you want to be taken seriously, is the scientific term for earwax. Anyway, the alleles come in wet earwax being dominant with the capital letter and dry earwax being recessive with the lowercase letter. And so you could have two dominant alleles, and that would be wet earwax. You could have two recessive alleles, and that would be dry earwax. Or you could have a dominant and a recessive allele, and that would be what now? Yeah, I'm listening. That's right, wet earwax, because you have a dominant allele, and a dominant allele is always expressed. There's some crumbly dry earwax right there. This is the dry earwax, two recessive alleles, and this is the wet earwax, and this could be two dominant alleles or a dominant allele and a recessive allele. You just don't know which because it's showing dominance. So let's get into some vocab to make it a little easier to refer to these gene pairs rather than two dominant alleles, two recessive alleles, a dominant and a recessive allele. That's a lot to talk about. So, homozygous dominant. Zygous, if we say things like zygosity, right, what we are referring to basically is genetics. Specifically, we're talking about the alleles and which alleles they have. So, some keys here. One, dominant. So we know that we're dealing with dominant alleles. So if it's earwax, there you go. And this prefix here tells you everything you need to know about the pair. So homo. Homo being Latin for the same. So if they are homozygous dominant, it is two dominant alleles. However, if you are heterozygous. Hetero is different. So if you are heterozygous, you have a dominant and a recessive allele, leaving only one other option, homozygous recessive. There's that prefix homo again, and it's telling us that they are recessive, so you have two recessive alleles, right? There we go. Not terrible so far. Now we'll go on to some more talkie-talkie and vocab here with phenotype versus genotype. Anyway, I underlined the PH in phenotype. So when we are talking about the phenotype of an organism, the phenotype, what we are talking about is specifically what is expressed, the physical expression. The physical expression. In the case of our earwax, it's either wet earwax or dry earwax. That's phenotype. Whereas when it's genotype, what we're referring to are the specific genes you have for that phenotype, the genes. And most specifically, the specific alleles that you are showing, the specific alleles that you have for that thing. So, in the case of genotype, if you're earwax, if we're doing with earwax, the phenotypes are wet or dry. In genotype, right, your possible genotypes are homozygous dominant, heterozygous, or homozygous recessive. Those are the three possible genotypes, right? So with our allele abbreviations, right, there's two big W's. Heterozygous would be a big W and a little W. And then recessive, two little W's. And this is important because you may show the dominant phenotype and not have homozygous dominant genotype, right? The dominant phenotype will have two possible genotypes associated with it, right? So you will have two different possible genotypes for the dominant phenotype. Whereas if you have the recessive phenotype, there is only one possible genotype. This is important. So, in this case, you have a homozygous recessive genotype, and your phenotype is dry earwax. And what's the phenotype of this little kitty? Short fur. And what's its genotype? Well, is it homozygous dominant or heterozygous? Which one? Which one? Oh, we don't know. You cannot determine genotype from dominant phenotype. If they show the dominant phenotype, you cannot know if it is the dominant genotype, or you cannot know which genotype it is, homozygous dominant or heterozygous. Okay, Now, as we get ready for talking about how to play with genetics and predicting offspring traits, let's talk about how sperm and egg combine to produce your specific genotypes. Right, so let's have Dad here, and Dad needs to make some sperm, and over here you have the mom, the biological mother, and she needs to make an egg, and so they might have their own specific genotypes. Here's Dad is, let's say, heterozygous, and just for consistency's sake, Mom is also heterozygous. All right? They need to make an offspring, a child. Right? And that child is going to have two alleles. It is not going to have both parents' allele set. If you are predicting alleles and you have like 4 alleles for an offspring, that is bad. No. Remember, the parents only donate one of their allele pairs. One. Okay, so the sperm only carries one of the alleles, right? So that sperm could have a dominant allele, or that sperm could have a recessive allele. And over here, the egg could have a dominant allele or a recessive allele. And which one will it have? It is random chance. Random chance. So we are talking a 50-50 chance of any specific allele from the pair winding up in the sperm or egg, depending on which parent it is. So, the parents for every gene have 2 alleles, and they give 1. Which one? Random. So, the gamete, by the way, is our fancy science term, and it is basically a sex cell. And that means that it could be sperm or egg. So if we say the male gamete, it's sperm. And if we say the female gamete, it's egg. Ta-da! So there we go. So let's make some babies. Oh, here we go. Dad being heterozygous, mom being heterozygous, the sperm could carry a dominant or a recessive, and the egg could carry a dominant or a recessive. So how do we know what the offspring possibilities are? Let's do Punnet squares, baby. The Punnet square, the best part of the genetics chapter, because it's fun. Dates between biologists always devolve into Punnet squares. And the one we're going to be concerned with is called a monohybrid cross. What is a monohybrid cross? It's where we are crossing for one category, right? So in a monohybrid cross, it is one category. So it is earwax. And if we're doing our good old human one. If it's cat, it's fur length, right? So it is not more than one at the same time, because there are crosses, like if we were to do a dihybrid cross, we would be doing 2 traits at the same time. So if it were cats, it might be something like fur color and sex. Why not? Right? So the fur colors, you'd have your alleles here, and for sex, you'd have your alleles there. And you'd combine them together into one cross, whereas here, you're only doing one category. Here we go, our Punnet square for earwax. So the most important thing for setting up a Punnet square is getting the alleles set up right. Like for setting up the Punnet square for a monohybrid cross, we have this square, right? With 4 different boxes. And to set it up correctly, we are going to want to first, right, we've got mom's genotype, she's heterozygous, put down what alleles mom can donate. And the answer is either one. So you're not, for instance, going to put this. This is a common mistake I see, where people put both of them over each column. No, bad. You put one option here and the other option there. So those are the females' donations to the egg, possible donations. Remember, it's a 50-50 random chance. And so then for the male's donation to the sperm, again, the male is heterozygous, so dominant or recessive. Once you have this set up, all you do is just combine the row and column for each square right here. So there we go. We have one option in which we are homozygous dominant. Over here, we have another option in which we're heterozygous. Over here, another option in which we're heterozygous. Whenever you have that, you generally just write the dominant allele before the recessive allele. So there we go. I have seen people write the recessive allele first and then the dominant allele. Nothing particularly wrong with that, but it can get wacky when they're like counting things up. If they put a recessive then a dominant, they might count it as different from this one because of the order. So if there's a recessive and a dominant together, just write the dominant one in first. And then over here, we are homozygous recessive. So these are the 4 possible outcomes. 4 possible outcomes depending on what allele the sperm gets and what allele the egg gets. Right? And this is probability, so, and because it's a 50-50 chance here and a 50-50 chance here, it's complete randomness, then basically it is a 25% chance of any of these outcomes occurring, right? It is easy because that means it is basically a 1 in 4, right? So a 1 in 4, because there are 4 possibilities, chance of any one of these outcomes. And that makes some interesting stuff we can say about probability. So if you have 2 parents who are heterozygous for a particular category like earwax, 2 parents heterozygous for earwax, there is what we call a 25% chance of being homozygous dominant. There is a 25% chance of being homozygous recessive. And there are 2 options where we have heterozygosity. So there is a 50% chance of being heterozygous. So we call this R the phenotypic probabilities. The phenotypic probabilities. So, phenotypic. Genotypic. Stupid me. Well, at least I didn't go on for like 40 minutes. The genotypic probabilities. And just like we have genotypic probabilities, we have the phenotypic probabilities, and it's pretty easy. The phenotypic probabilities are just looking at how many options give each different phenotype. So we have a 25% chance of homozygous dominant, and for our earwax, that means wet earwax, right? Wow. Dominant is wet earwax, and recessive is dry earwax. So homozygous dominant, wet earwax, right? And looky here, heterozygous, you've got a dominant allele, and if you've got a dominant allele, you're the dominant phenotype. So wet earwax and wet earwax, 3 out of 4 options means there is a 75% probability that any given offspring from 2 heterozygous parents for earwax would show the wet earwax phenotype, and only a 25% probability that any offspring would show the dry earwax phenotype. So we have the Punnett square. Setting it up is important. Remember, when you set it up, your offspring, all the offspring probabilities should have, 2 alleles. If you set it up and you're getting 4 alleles, you did something wrong. If you set it up and they're getting 1 allele, you did something wrong. So setup is very important. So just remember that the parents can only give 1 allele from their pair. And if they're heterozygous, it's 50-50 which one it could be. Right? Here's dad's heterozygous. And so 50-50, you're just essentially slapping your sperm down here and your eggs up here. It's the sperm options and the egg options. But in case you wanted to see it all PowerPoint magic-y, boom. Right, there's the phenotypic. Right, we have 3 phenotypes that are wet, 1 that is dry, 1 phenotype that is, or 1 genotype homozygous dominant, 2 heterozygous, 1 homozygous recessive, and from there we can determine the probabilities. Right, these are not guarantees. If they have 4 children, you will not get 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. These are probabilities. So there are 4 possible outcomes from a Punnett square. In this case, 1 in 4 is the homozygous dominant. So there's a 25% chance any offspring would be homozygous dominant. 2 out of 4 is a 50% chance that any offspring would be heterozygous, and a 1 in 4 that any offspring would be homozygous recessive. And the phenotypic ratios, boom. Okay, so pedigrees. Pedigrees are when we track these traits across multiple generations. So we are trying to look at how these traits have been inherited through more than one generation, more than just parent to offspring. So it'll be like a family tree. And we want to just look at the map of inheritance in that sort of family tree, that pedigree. And it's pretty easy, right? So what you do is you set it up. So here's the male parent, calling him dad. And here's the female parent, calling her mom. And when we set up in our map in our graphical representation, we are generally setting up with phenotypes, right? A pedigree is generally phenotypic, most often because, quite frankly, not everyone wants to do a bunch of genotyping for multiple people across multiple generations. All right, so we will use squares for the biological males and circles for the biological females. And then for the phenotypes, we will indicate that by either having a square colored in which case it is the dominant phenotype, or the square circle being empty, in which case it is the recessive phenotype. So filled in is a dominant phenotype, and not filled in is a recessive phenotype. So here we go. For earwax, right, dad is a square that is filled in. So what phenotype is that for earwax? I'm waiting. Come on, look backwards. Flip through those papers. The dominant phenotype, right? Wet earwax. And here's mom, right? Circle to show that it's a biological female. And the phenotype is recessive, so this is dry earwax. One parent has wet earwax, one parent has dry earwax. And they have 3 separate offspring. 2 males, 1 female. And we can see both males here are showing the dominant phenotype, but the female offspring is showing the recessive phenotype. Ooh, fascinating. How could we have one parent showing the dominant phenotype and one parent showing the recessive phenotype have offspring that is recessive, right? Don't we have the dominant thing going on here? What is happening? Well, it helps to think of it in terms of alleles. Remember, right? Our specific alleles. We have the dominant allele, coding for wet earwax, and the recessive allele, coding for dry earwax. And we know that each parent has 2 alleles. And we know the specific allele combinations for mom. Because she's the recessive phenotype, she must have 2 recessive alleles. You can only show the recessive phenotype if you have 2 recessive alleles. And we also know the genotype of that female offspring, because again, that female offspring is showing dry earwax, recessive phenotype, and we can only show that if we have 2 recessive alleles. So, we know that each offspring gets 1 allele from mom, 1 allele from the female parent, 1 allele from the male parent, right? We know that the male parent has at least 1 dominant allele. And we know that each of these male offspring has at least 1 dominant allele. They got it from their male sperm contributing parent, right? Now, we know mom can only donate a recessive allele. So all these offspring have 1 recessive allele for mom. So there it is. We know these 2 kids' specific genotype. We know this kid's specific genotype. So the question is, if this one is from the egg, where is this one from? It has to be from the sperm. So we know that the female parent is homozygous, recessive, We know the male parent is heterozygous. We know that each male offspring is heterozygous. And we know that the female offspring is homozygous dominant. Or recessive. Getting away from me. Thinking ahead. All right, so this is pretty simple. This is an ideal pedigree right here for figuring out genotypes. I like a pedigree like this because if one parent's homozygous recessive and one offspring is homozygous recessive, then based on the phenotypes of the other offspring and the other parent, you can pretty much genotype out everyone using this pedigree. It's nice. I like it. However, if both parents had the dominant phenotype and all the offspring had the dominant phenotype, circles are easier to draw. Well, you would not be able to tell any of the genotypes, right? If both parents had wet earwax and all the offspring had wet earwax, then you would honestly have no idea. Because remember, with a wet earwax, you can either be homozygous dominant or heterozygous. And because we have no other possible options to narrow this down for us, we have no idea what any genotypes are for anyone in this pedigree. So bear in mind that for some pedigrees, you can work backwards or forwards and figure out everybody's genotypes. For some pedigrees, you honestly cannot. Right? And some you can get partial information out of it. Right? So if both parents have the dominant phenotype and they have two offspring, one with the dominant phenotype and one with the recessive phenotype, you actually can chart out some genotypes, right? This is homozygous recessive. And the only way this could be homozygous recessive is if we get a recessive allele from each parent. So each parent must have a recessive allele, but because they are the dominant phenotype, they also have a dominant allele. So both parents are heterozygous. One offspring is homozygous recessive, but right here, right, this offspring, it could have received a dominant allele from the sperm and a dominant allele from the egg, or it could have received a dominant and a recessive allele from either one. So you do not know. You cannot tell with certainty what this offspring is. So pedigrees can give you different amounts of information for genotypes depending on how they're set up. Carrier is some important vocab to learn. Carrier, it means you carry an allele but don't express it. You carry the allele, but you do not express that allele. You don't express it. Basically, in what we've learned so far, that's recessive alleles. You are a carrier for a recessive allele if you are heterozygous. Heterozygous are carriers for recessive alleles. Heterozygotes are carriers for recessive alleles. So, and then offspring 1 and 2 are also carriers, right? Because we were able to work out all the genotypes. So all of the offspring that are displaying the dominant phenotype, like both offspring displaying the dominant phenotype, and the parent, the male parent, are carriers. They're all carriers. So, boom. One is actually displaying the recessive trait. One offspring is displaying the recessive trait. Someone who is expressing a recessive trait is not a carrier. By definition, carriers do not express it. So you can only be a carrier if you're heterozygous. So let's move on to some basic genetic disorders. Some genetic disorders show this very simple inheritance pattern, this dominance recessive inheritance pattern. Some are nice and simple, right? Dominant or recessive alleles. Some disorders are more complicated. Let's go through some simple ones. All right, recessive genetic disorders. This right here are some pretty funky looking red blood cells. So here's some red blood cells. Red blood cells. The notorious RBC. Anyway, this is a pretty normal looking red blood cell. And so is this one, and so is that one. These are pretty nice looking red blood cells. But what is going on here? What? This is a messed up, freaky looking red blood cell. It almost looks like, oh, I don't know, a sickle? Huh? Oh, sickle cell anemia is our first recessive genetic disorder that we will talk about. So, sickle cell anemia is where you have malformed hemoglobin, right? You remember hemoglobin, right? Primary component of hemoglobin is the protein heme, and heme carries iron, and iron, right? Iron attracts oxygen. So hemoglobin is the oxygen carrying structure in red blood cells. And in sickle cell anemia, your hemoglobin is malformed, and that causes these weird malformed red blood cells. They call them sickle red blood cells. This is not a great disorder. Sickle red blood cells are mildly dangerous in small amounts and very dangerous in large amounts. And the reason for that is the way red blood cells move through your smallest blood vessels, the blood capillaries, right? So these are the smallest ones. And if you have a normal red blood cell, it is kind of a little bit larger than a capillary. And so it squeezes down, it bends as it goes through the capillary. If you have a sickle red blood cell, it doesn't bend as well. And more than that, it's pointy, and it can cause damage as it passes through capillaries. So sickle cell will often lead to kidney damage. It will often lead to liver damage, and it can also destroy the capillaries in your extension in your hands and feet. So you can end up with the tissues in your hands and feet dying, and you may have to get it cut off. So not great. These sickle cells also carry less oxygen, as you might imagine, and so that's where this anemia part comes in. that anemia, basically you are not carrying enough oxygen on your red blood cells. You become anemic, and that leads to exhaustion. You have exhaustion, you have low energy levels, not great for you. So, damages the spleen, you get this anemia, not awesome. So, if I were going to show this, right, people will have the sickle gene or not. I don't like S as an abbreviation because, you know, the lowercase and the uppercase are virtually identical. So, sickle cell, sickle. Let's just go with the K. Not ideal either, but yeah, there's nothing real good in there. We'll just use the L because why not? There's an L in there. So you might have the dominant not sickle allele or the recessive sickle allele. Right? So most people, not most people, but I mean, if you are a white person living in America, you are likely homozygous dominant. The probability is high that you are homozygous dominant. Then you have the people who are heterozygous, and they are carriers. And then you have people who are homozygous recessive, and they are full-blown sickle cell anemics. And it is not great. If you have sickle cell anemia, you pretty much have to get regular blood transfusions in order to eliminate the sickle cells from your blood vessels. So it is not the most convenient treatment in the world. But it is nice that you can be treated with blood transfusions. It is no longer a death sentence in modernized healthcare. So that's nice. I mentioned white people partly because there's something super cool going on in sickle cell anemia. Um, and that is something called, well, you're not going to get tested on this, but sickle cell anemia has what we call heterozygote advantage when it comes to inheritance. So, I'm going into a little bit of a sidetrack here. Heterozygote advantage. What is heterozygote advantage? Well, I mean, it deals with inheritance, and basically it means that there is an advantage to inheriting heterozygosity. Okay, and this is all about malaria. I'm going to get to a, let's see, can I just erase this? I know there's a little menu in here. What do we got here? Give me this. Erase all ink on slide. Okay, so heterozygote advantage. What is that? This is all about malaria. In countries, like specifically equatorial countries that are hot and humid, malaria is a big problem. Malaria is this like little blood-borne protozoan. It's annoying looking. Like, here we go. Yeah, I'm an evil little protozoan and I'm going to attack the cell. Ha ha ha. And it forces its way into the cell, and then it gets a bunch of these little it reproduces itself into a bunch of little things that fill up the cell and and it keeps reproducing and it keeps reproducing and it keeps reproducing until eventually there's about as much as can fit in here, and then kablooey, they erupt from the cell, and that cell is most certainly a dead cell, right? Malaria is very deadly. It is a lethal disease. It's mosquito-borne. Um, Basically, if a mosquito feeds on someone carrying malaria, infected with malaria, the mosquito sucks up a bunch of these little things, and these little things can survive in the gut of a mosquito and then migrate to its salivary glands so that when it drinks the blood of another person, they'll get out in the saliva. So, pretty scary. Anyway, this is a very dangerous, dangerous disorder. Um, if you get malaria and you don't have access to modern health care, there is a pretty high chance that you are going to die. All right, but check it out. If you are the little malaria plasmodium being all evil and wanting to infect this blood cell here, uh-oh, what? What's up with this weird sickle cell? I can't get in there. I can't reproduce. Sickle cells are immune to malarial infection. And this gets us to heterozygote advantage because if you are heterozygous for sickle cells, right here, carrying that recessive allele means that you have some sickle cells. But most are normal red blood cells. Some are sickle red blood cells. Most are normal red blood cells. And thus, if you get malaria, right, well, first, you have mostly normal red blood cells, which means that you do not have the anemia part. No anemia, you're mostly fine. But some sickle red blood cells means that if you get a malaria infection, it is much less likely to be lethal because you have these red blood cells in your body that are immune. This is super cool, right? So generally, populations descended from humid, hot equatorial countries are disproportionately heterozygous. So um, the African American populations in the United States show a much higher rate of heterozygosity for sickle cell than the white populations. Which I always thought was pretty cool because that means you are malarial resistant. It is your superpower, which is pretty sweet. When the evil genius releases malarial mosquitoes into the city, you heterozygotes will be able to fight back. I don't know. Anyway, moving on. There are dominant genetic disorders as well. There are no carriers for dominant genetic disorders. Why are there no carriers for dominant genetic disorders? Why? Come on, look it up. You just wrote it down just a little bit ago. Come on, go back. Go back. What is the definition of a carrier? By definition, a carrier is possessing an allele but not expressing the allele. But in a dominant genetic disorder, the disorder is carried on a dominant allele. And a dominant allele is always expressed. A dominant allele is always expressed. So there are never carriers for a dominant genetic disorder. One example of a dominant genetic disorder is good old osteogenesis imperfecta. Where did my gecko go? I'll have to figure that out now. Hi. One of your disorders is Osteogenesis Imperfecta. Osteogenesis Imperfecta is a disorder where one of the proteins that are very important in making up your bones, it's called collagen. We'll talk about it a bit later. But your collagen does not form correctly, right? And when your collagen doesn't form correctly, the bone doesn't form correctly. And that's very dangerous because collagen gives bones strength and flexibility, right? The calcium part of your bones, right? The calcium crystals that are part of your bones, they provide your bones rigidity, so that they provide a nice rigid structure for muscles to attach to. But collagens provide strength and flexibility. So if you do not have collagen protein, if you have defective collagen protein, then your bones they don't generally form correctly, and they break extremely easily. So you have extremely fragile bones that break very easily, and they generally are like the support bones, these long bones, they're generally bent because they don't they don't support the body as well. So you get these bone deformations here because they are defective. Yeah, and they're not working really well. So anyway, the kids, they called me Mr. Glass. Right? That suddenly relevant again after the sequel, Glass, that came out. Woohoo! That reference was very irrelevant for a while. Completely irrelevant for a while, and then it became relevant again. Celebrate with me, Gecko. Dance. Dance. Anyway. So you have defective collagen, reducing your flexibility, meaning you have brittle, weak bones. Now, this is treatable in modern healthcare. You are given drugs, significant drugs, to increase collagen production. Right? Your collagen may be defective, but if you can crank it out in high amounts, you can decrease the severity of the disorder. You can get something near normal bones as long as you can overproduce that collagen. Another dominant disorder is achondroplasic dwarfism. Yay! Achondroplasic dwarfism is pretty cool. Well, I don't know. Whatever. Genetically speaking, it's a neat disorder. I should not be as enthusiastic. It makes me sound weird. So, in achondroplasic dwarfism, you have short limbs that tend to be disproportionate compared to the torso. Not all dwarfism is equal. Like the people in the original Wizard of Oz movie, the lollipop kids, right, of Munchkin Town. Right? All those people were dwarves, but they had a growth hormone disorder. So they didn't produce enough human growth hormone. And the key there was that they were all in complete proportionate, their limbs were all completely proportionate to their body. So they were not achondroplasic. Achondroplasia, you know, from Game of Thrones, right? Peter Dinklage has achondroplasia. So, yeah, generally you're 4'10" or shorter, and you have those shorter limbs. How does inheritance of achondroplasia work when you're a heterozygous? So if you have achondroplasia, right, here's your dominant allele, and here's your recessive allele. You can donate either the dominant allele or the recessive allele to your gamete. So Here's someone with achondroplasia. Here's someone who doesn't have achondroplasia. We'll just punnet that baby out. And so your probabilities are 50% that your offspring will have achondroplasia and 50% that your offspring will not have achondroplasia. Now, it is worth noting with achondroplasia that just about everybody who has achondroplasia is heterozygous. Achondroplasia is primarily, pretty much everybody. I'm not going to say all people with achondroplasia because there's probably an exception somewhere that would make me wrong. But the thing is that the homozygous combination, if you are homozygous dominant for achondroplasia, you have two dominant alleles. This is a lethal combination. This is what we call lethal achondroplasia. I don't know the specifics about the mechanism that causes it to be lethal. I know most fetuses that have it don't go to term. I don't know if you get offspring born with lethal achondroplasia, but it's pretty much, it is lethal. So, if you see someone like Peter Dinklage, he is almost guaranteed to be heterozygous for achondroplasia. One of the cool things about inheritance in achondroplasia is if you have two parents that are heterozygous for achondroplasia and you punnet that out, you will have, there is homozygous dominant, there's heterozygous, and there's homozygous recessive. You have three possible ways of showing the achondroplasia trait. So there's a 75% probability that you will have achondroplasia and a 25% probability that two parents with achondroplasia will have offspring that are not showing achondroplasia, that do not have achondroplasia, which is pretty cool. Now, of that 75%, there is a 25% chance that you will see lethal achondroplasia, which ain't great. So, but there you go. It's always just one of the neat things out there that if two parents have achondroplasia, there is a 25% chance that any offspring they have will not show achondroplasia. So that's pretty cool. Peter Dinklage himself has a wife who does not have achondroplasia and a child that does not have achondroplasia. But Peter Dinklage has achondroplasia. So if we punneted that out, or first we'll pedigree it out, just because we learned pedigrees and it's fun. She's just making a weird, funny face, and the photographer took a picture, and it looks like unflattering, but very cute. Anyway, here we go. Here's our pedigree. We have Peter Dinklage with achondroplasia, his wife not having achondroplasia, and his daughter not having achondroplasia. And we can know offhand that these two are homozygous recessive, and aside from the fact that Peter Dinklage is alive, we see that the daughter has 2 alleles for not having achondroplasia. Mom can only donate 1, so Peter Dinklage must have the other. So Peter Dinklage is heterozygous for achondroplasia. Pretty sweet. Genetics is fun. Here we have 2 parents that do have achondroplasia. Where'd my cursor go? There it is. 2 parents with achondroplasia and 2 offspring with achondroplasia and 2 offspring without achondroplasia. So this is a pretty standard illustration, or this is a pretty good illustration of probability as well, because remember, it was a 75-25 when it comes to achondroplasia or not. And so with 4 offspring, if those rules were guaranteed, you would only have 1 being not showing achondroplasia. So it's all random numbers. So now let's talk about when it gets weird, baby. So this stuff will not be on the exam, so that's just because I got super lazy. By the way, this is referencing spring 2019 specifically, in case any of you are not in spring 2019, and I'm choosing to be a dick to you. So for you spring 2019 folks, this non-mendelian stuff, this non-simple inheritance stuff is not on the exam. So let's talk about when inheritance does not follow the rules. So we have several options here. This is not exhaustive, but we have polygenic. Might be able to figure out from the word poly and gene, but anyway, we'll get onto there. A subset of polygenic is multifactorial. We have pleiotropy, and then incomplete dominance, and another type of inheritance called codominance. And finally, multiple allele inheritance. So let's go on. Polygenic traits. Again, poly being the Latin prefix for many, and gene, so there is more than one gene governing this trait. One trait, many genes. The gold star example of this is skin color. Skin color is an absolutely beautiful example of polygenic traits. There are 3 genes governing skin color, and your specific skin color is just the sum of dominant and recessive alleles. So the intensity of skin color is based on how many dominant alleles you have. So with skin color, you have dominant alleles producing melanin. Melanin is a skin pigment, and it absorbs, or if you like to think of it, blocks ultraviolet radiation. It absorbs or blocks UV. Melanin is protective, which is pretty sweet. It is protective. Okay, so darker skin colors have more melanin. So for these 3 separate genes that govern skin color, all of them are dominant, producing melanin, and recessive, not producing melanin. That's literally the only genetic factors governing skin color. That's it. There's nothing linked to it. If someone else tries to tell you there's anything else associated with skin color besides melanin, slap them upside the face. Note, these views are not considered expressive of the views of the university that I am a representative of. I am not a representative of that university's views or opinions on whether or not you should commit violence against people. And you probably shouldn't commit violence against people. Anyway, moving on. So, the intensity of these traits is based on the number of dominant alleles, right? So you might have gene A, gene B, and gene C. And if you are heterozygous, whatever, I'll just use A's. So if you're heterozygous for A, heterozygous for B, and heterozygous for C, your cells will produce sort of 50% of the maximum amount of pigment a skin cell could have. Right? If you are homozygous dominant for all three genes, your skin cells will produce the maximum amount of pigment that any particular skin cell could produce. So you have this beautiful variation. If you're homozygous recessive for all three, you will produce essentially the smallest amount of pigment. Note, important note, this is not albinism. People who are homozygous recessive for all three genes are not albino. There is a specific gene for albino. There's a specific gene for albinism. And if you have the albino gene, don't quote me on this, I think it's just a dominant recessive thing, but it's interesting. It's a different kind of inheritance. Basically, with that gene, if you do not have albinism with your particular allele combination for the albino gene, then these genes do their thing. However, if you have the allele combination that results in albinism, that allele combination causes this to be ignored. All three genes are completely ignored, and the albinism alleles are what are expressed. So in that case, one gene can block the expression of other genes. And that was a huge tangent. I'm not even technically talking about that kind of inheritance right now. So anyway, it's pretty sweet. Skin color, three genes, dominant is melanin. More dominant alleles means more melanin. And it produces this beautiful, beautiful gradient of skin color. And what's fascinating is there's also a want to say latitudinal variation here. So at the north and south, as you go towards the north and south pole, you tend to see these much lighter skin colors. And then as you move towards the equator, you see these darker skin colors, right? So here's your Earth, here's your northern and southern poles, and you tend to get around the equator these darker skin colors. And the reason has to do with good old sunlight. I shouldn't draw it up here. That's like how we draw it when we're in grade school and drawing it. Right, so here's the big old sun. There's the corona. Anyway, it casts light at the little bitty Earth, and that light comes in, and it hits the Earth, and it comes in at an angle, and as you get towards the poles, light is deflected away. Right, so less light reaches the poles. Very little light reaches the poles. The intensity of sunlight at the poles is much, much lower than the intensity of sunlight at the equator. Now, humans have to produce vitamin A. And the catalyzing agent for the production of vitamin A is light. You can end up with a deficiency if you don't get enough light. However, with sunlight comes UV radiation, and UV radiation is damaging, right? It damages your cells. The more UV radiation you have, the higher your chance of skin cancer because it's damaging your cells. So, there we go, UV radiation. Bad. Light for vitamin A, good. So, your body must govern how much light is absorbed past the skin cells to get vitamin A production versus how much UV you are exposed to. Melanin is protective against UV. So where the light is most intense, where you do not have any problem at all getting enough sunlight for vitamin A, it is a very good advantage to have lots and lots of melanin. So evolutionarily speaking, the equator selects for melanin production. And as you get further from the equator in either direction, less light comes through the atmosphere, you get less intense light, and you need less melanin if you are going to get enough light to produce your vitamin A. So there you go. That is why you often see that distribution of pigment based on how near or far you are from the equator. Pretty sweet. Okay, multifactorial traits. This is where you have polygenes and the environment directly affecting whether or not a product is made. So, in this case, our example is Himalayan rabbits, which are pretty cute. There's a little ear, and there's a little ear, and there's my little bunny face, my little bunny nose, there's my little bunny teeth, right, and then there's my little bunny feet, there's my big old bunny feet, and there's my little bunny butt and my poofy bunny tail. Alright, so Himalayan rabbits. A Himalayan rabbit has what we call dark points. Patches of black fur on the tips of their ears, and oftentimes their entire ears. Their little bitty bunny nose, right? Their little bitty bunny feet, their bunny toesies, and their poofy bunny butt. They have what we call black points on Himalayan rabbits. And again, this is melanin. Okay, now, with Himalayan rabbits, these points are black only in specific environmental conditions. So, this Himalayan rabbit has the genes, all their cells, every single one has the genes to produce this much melanin. So why isn't the entire rabbit black? Why is a Himalayan rabbit white except for black points? Well, that's where the environment comes into play. Because melanin is protective against UV, but if you own a car and in the summer you get in your car with its black interior, you cook to death and burn and die. And that is because black absorbs heat really efficiently. Black is basically, the color black occurs when light is absorbed. All the wavelengths of light are absorbed to produce the color black. And so that is why it absorbs heat very well, because light carries heat. And that's where we get to bunny. Because think about where heat loss occurs, right? Their little toesies are what are in contact with the ground. And the ground can be cold, and they can lose heat. Their poofy little bunny butt tails, right? Those are sort of exposed on their own. Their ears, exposed. Their little nosy, where they breathe in cold air, exposed. These are the sites of heat loss. The points are sites of heat loss. The rest of our bunny, give it an adorable little eyeball. The rest of our bunny is this nice insulated with muscle body here. And it's got all this beautiful insulation, all this warmth carrying through it, so it doesn't get that melanin to absorb light and reduce heat loss. The black points reduce heat loss. So, because our points are sites of heat loss, that black color from the melanin reduces heat loss. Great for our Himalayan. And that means it's literally temperature, which is the environmental condition that determines expression of this polygene trait. Oh, so there were actually studies done where scientists took a fully grown Himalayan rabbit and they shaved its back, and then they strapped an ice pack onto its back so that they kept that back cold, and when the fur regrew, it was a black fur. So the environment determined the expression, even after maturation, even in adulthood. The right environmental conditions determined that expression. Pretty cool. So they have the homozygous genes, or they have the genes C and H, and if they're homozygous dominant, if they're homozygous for melanin production for both C and H, they produce a whole bunch of melanin, enough to get that deep black fur, but those genes are inactive. They are not turned on unless you have heat loss. And yes, you have reduction in temperature. So, low temperature. Here's them adorable little bunnies. Oh my god. So cute, little fuzzy bunny nose and their ears and their cute little toes. Oh my god, look. His puffy, poofy little bunny tail. Okay, anyway. It's heat loss. That reduction in temperature activates the CH genes. Pretty sweet. And this is that scientific experiment where they shaved that bunny, strapped an ice pack on while the hair regrew, and once the hair had fully regrown, it was black fur. Okay, on to Pleiotropy. This is where you have many traits governed by one gene, sort of the opposite of polygenes. In polygenes, it was one trait, fur color, skin color, governed by multiple genes. In pleiotropy, it's multiple traits governed by one gene. And these traits often seem to be unrelated. Right? So in humans, Marfan syndrome is an example of a pleiotropic disorder. It is dominant, it is inherited, as a dominant disorder. But unlike our simple dominance inheritance, where it would be affecting one thing, in pleiotropy it affects many things. One allele, one dominant allele, and you have disproportionately long arms, legs, fingers, poor eyesight. Well, that doesn't seem very related to limb length. A weakened aortic wall, heart versus eyes versus limbs, and other, my favorite expression of genes. Anyway, this is a kid with Marfan syndrome, with his disproportionately long limbs. So it affects, and disproportionately long fingers. It affects a lot of cool stuff. So there's actually, in horror movies, if you see an actor, there's this one actor, I can't remember his name, but he actually made a name for himself by being super duper creepy in horror movies. And he's able to pull it off because he literally has Marfan syndrome and has super long arms, legs, and fingers compared to his torso and head. And so he looks, you know, kind of freaky with the right makeup. Okay, incomplete dominance and codominance. We're getting closer to the end, folks. I promise. All right, these are two separate things. They seem kind of close in concept. Okay, so with incomplete dominance, you have a situation. It's back to one pair of alleles, one trait. Ah, so it seems like it might be, uh, or one category. It seems like it might be dominance recessive, except for a fundamental difference. In incomplete dominance, neither allele is dominant over each other, and expression is a blend of those two alleles. So a classic example might be in some of those snapdragons that you get from the flower store, right? So white snapdragons and red snapdragons, very pretty, right? So R is a nice big difference between the letters there. Or you know what? We'll just use. There's no dominance recessive, so I don't need to worry about capital versus lowercase. Okay, so here's the red allele. Here's the white allele. A white flower, a white snapdragon, has two white alleles. A red snapdragon has two red alleles. And a snapdragon that is heterozygous for red and white has a red allele and a white allele. If this were dominance, they would be either red or white. This is incomplete dominance, which means neither of these are dominant over each other, and importantly, they are a blend. So when you blend red and white, you get pink. So with these particular snapdragons that you buy from the grocery store, the pink snapdragons are heterozygous for the red and white allele. There's snapdragons of multiple different colors, so I don't think all colors in snapdragons are governed by these alleles. They can't be. But for these red, white, and pink snapdragons, the pink are a heterozygote. They are a blending, a blend of the alleles. Yeah, I made such a big deal about this because basically your alleles are averaged, right? So sickle cell technically comes in incomplete dominance because you have some sickle cells and normal red blood cells. So technically, you're getting uh, multiple kinds in there. Anyway, codominance. Okay, in codominance, neither allele is dominant, right? So you don't have one allele covering the expression of the other allele. But the big difference here is both are fully and completely expressed. Both alleles are fully expressed. Blood type is your classic example of codominance, right? Some people might be blood type A. Some people might be blood type B. And some people might be blood type AB. I'm going to go into a little more detail because blood type's a little more complicated than this, but this has to do with your little red blood cell and little receptors on it that are like identify friend or foe, right? Your immune cells, those macrophages I talked about, whose whole job is to eat the dangerous stuff, they will bind to red blood cells. They'll move along, come into contact with a red blood cell, and check out these proteins that are on the surface, right? And you might have A proteins, like this, in which case you have the gene for A proteins. So you have the A allele. And you might have B proteins. Right? So your immune system rolls around and it checks out your red blood cells. And if you have A proteins, that means A proteins are yourself, so they're good to go. If your macrophage attached, if you were type A blood and your macrophage attached to some B proteins on there, it would kill that red blood cell. It would attack and kill it. Anyway, here's the B allele. And if you have one A and one B allele, you fully express A proteins and you fully express B proteins. You fully express them. If this were some kind of incomplete dominance, then it would be some sort of blending of the A and B proteins, making some weird C protein. But it's not. You fully express them. So, snapdragons, pretty. Aw. And here's horses in codominance. And this has to do with roan horses. So you have horses that have white hairs and horses that have red hairs that kind of look like a rich brownish reddish. So if you have a horse homozygous for the white hairs and a horse homozygous for the right hairs, and you homozygous horse for white hairs and a homozygous horse for red hairs, and you breed them, they will get one allele for white, one allele for red, and they'll be roan. And you will get hairs that are either fully white or fully red. And roughly 50% of the horse's hairs will be white, 50% of the horse's hairs will be red because they're expressed fully. So this is a white horse. Where'd my cursor go? Here's a white horse. Here's a red horse. And boom, this is a roan. And it looks almost like a blend, but if you look closely, when you look at a lot of these individual hairs, especially looking at the white hairs here, you see those white hairs are pure white hairs. and the red hairs are purely red hairs. So we are fully expressing white hairs and we are fully expressing red hairs. So very different from blending. And now we'll really get into blood type here with multiple allele inheritance. Right? Because up until now we've learned that you have one category. Right? You might have one category, and that category would be like our earwax. Right? Right, here's your earwax. And you had an allele for dry, and you had a possible allele for wet. And that's it. In multiple allele inheritance, you have one category, and I've already given away that it's going to be blood type. One category, which is blood type, however, you have more than two alleles. A alleles, B alleles, and that third one, O alleles. Right? Three different alleles. You have more than two. Multiple allele inheritance is where one gene in the so let's specify this so it's not confusing. In the population, the breeding population, you have at least three different alleles. Your genotype is still based entirely on the allele pair. You can only have two alleles, but in the population floating around, there are three or more variations. In blood type, in the population of humans, there's alleles for A, alleles for B, and alleles for O. With earwax in all humans in the population, you either have wet or dry earwax alleles. But in blood type, in the population circulating around are three different allele options, specifically written out as capital I with a capital A there, a capital I with a little capital B, and then a lowercase I. Lowercase does not mean recessive. It's just how they wrote it out. So here we go. These code for different things. I've already given it up that these are those cell proteins. So the A allele codes for A protein on the red blood cell. The B allele codes for B protein on the red blood cell. And the O allele does not code for a protein. Not coding. So it doesn't code for any particular protein. So this means you can have a lot of different allele combinations in the population. Right? You can have combinations, any pairs from all three different ones out there. So you could have people who are IA, homozygous for A. You could have people who are homozygous for B. So homozygous for the A allele, homozygous for the B allele. And you could have people who are homozygous for the O allele. Then we'll continue on in our combinations. You could have someone who is heterozygous for A and B. And then you can have people who are heterozygous with the O allele. So you have a type A heterozygote, heterozygous for A and the little I, and heterozygous with the B. So there we go. These are our different genotypes all out there in the population. Right? That's a lot of different genotypes floating around. 1, 2, 3, 4, 5, 6 different genotypes floating around out there in the population. And they are associated with multiple different phenotypes. Some extras considering that this is codominant. So if you have either of these, you are blood type A. So if you are homozygous for the A allele or heterozygous with the A and a little I, here you are type B. Here you are type AB. And here you are type O. Now you will notice I am not talking about positive or negative. Right? We all know type O positive, type O negative, whatever. This set here has nothing to do. The genes that govern the letter A, B, or O, or AB, that is actually completely different from whether or not you are positive or negative in your blood type. It's a completely different set of proteins, too. So, here we go. If you're type A, you might have those little triangles, right? Because this is codominance, you have, for when you're homozygous for A, you're fully expressing this one with A and fully expressing this one with A. Makes sense, right? But here, you're fully expressing the A and fully expressing something that doesn't code for a protein, which means you're fully expressing the A. So both of those are type A. Here, type B, right? I use my little squares. Right here, when you're homozygous, you're obviously fully expressing a B and a B, so you're B. Here, when you're heterozygous, you're fully expressing a B and fully expressing something that doesn't code for anything, so you're fully expressing a B. Here, you are carrying an A allele and a B allele, so you fully express both of them. And only down here, when you carry two non-coding alleles, you have that little naked red blood cell that's not coding for any of those. So that is how multiple allele inheritance works. In the population, you have more than just two alleles floating around. Red blood cell or blood type is a great example of that because everybody has some passing familiarity hearing AB, or type O. Uh, so, um, nice. Uh, Tangent, real quick tangent. The immune system will attack anything that is not you. Right? It detects these red blood cell proteins and attacks them. If something's detectable by the immune system, it's an antigen. Right? An antigen is anything that can be detected by the immune system. Not everything can be. So these are proteins. These proteins are all antigens. And so if you have type A blood, that means your red blood cells are carrying type A protein. Thus, type A protein is you. And your immune system will attack any cell, any red blood cell that isn't you. It's a self versus non-self system. So if you get a transfusion with type B blood, your immune system rolls around and hits this blood cell and goes, yay, that's me, and hits this and goes, whoa, what the f- is this? No, thank you, and attacks and eats and kills it. And if you get type AB in your type A, it says, whoa, it's like, oh, well, this is cool, but hold up, buddy, no, 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 and attacks and kills those. And if you're type A and you get a transfusion from type O, your immune system rolls around, hits some of these that are you, and says, hey, type A, that's me. And it goes, nothing to see here, folks. Move along. This is ninja blood. Ninja blood cells. They do not carry the antigen, so if they don't have the antigen, they're not detectable by the immune system. So type O. Right? If you have type AB blood and you get a transfusion from someone with type A blood, your immune system is like, oh, hey, this is me, and it comes along and it says, oh, hey, this is me, and you get type B blood, and it's, oh, hey, this is me, and you get type AB blood, well, this is me, and you get type O blood, this must be me. So, if you have type AB blood, congratulations, you can just suck out anybody's blood for a transfusion. Yay! As far as ABO blood groups go. This is the ABO blood groups. This is because positive and negative is an entirely different type of blood group. This is the ABO blood group. Right. I might even take a second to talk about it. Anyway, so genetic pools 3 or more alleles. You only get 2. Blood type ABO. There we go. Did all that, but much more attractively. This is a terrible internet thing. I'm better. Uh, there's the immune system. It's an engine for attacking self versus non-self. It attacks anything that's non-self, and these proteins are an antigen. So those red blood cell proteins, A and B, are red blood cell proteins that can be detected and reacted to by the immune system, and thus are used in determining self versus non-self. So your specific combination is you, and that is yourself, and those are the red blood cells that get a pass. By the way, I'm type O, which means this is self. So if I get a transfusion from AB, my immune system goes, what the crap is all this? And attacks it. And same with A, and same with B. I can literally only get transfusions from someone who is type O. Ain't great. So any other combination is, if you don't have your specific combination, is non-self, but it's because it's codominant, there's sort of some nice stuff. One last wrinkle, well, you know what? For a moment, why don't I digress real quick? Because this wouldn't be on an exam, but uh, I've talked about blood type, and some of you are probably curious. So let's talk about what we mean when we say positive or negative, right? This is not the ABO blood group. In fact, this is something that we call the rhesus blood group, named after the fact that they detected it in the rhesus monkeys. And this is, once again, a red blood cell protein that is an antigen. Another antigen. Now, the good news is that this one, well, good news, whatever. What's neat is that this one is literally just the simple dominance and recessive that we've talked about. Simple dominance and recessive. It's literally just like earwax, and it goes like this. The rhesus protein, can either be present on the red blood cell, or it can be absent on the red blood cell. If it's present, we call this Rh positive, and if it's absent, we call it Rh negative. So, since this is triangles and squirrels, since this here, in this little image, which I'll be lazy and just use, type A is triangles and type B is circles. So I'll just make my rhesus the squares. So rhesus positive would be having the rhesus protein. So this person is going to be type A positive. They have the rhesus protein, and thus the rhesus protein is one of the antigens that means itself. And this person is type B negative. Right? So when doing stuff like this, we often use plus minus for our alleles. So plus is the allele for Rh positive, for having the rhesus protein, and minus is the allele for Rh negative, for not having the rhesus protein. So being simple dominance recessive, your combinations could be homozygous dominant, heterozygous, and homozygous recessive. 3 different possible genotypes and 2 different possible phenotypes. In here, you are positive because the positive allele is dominant, and here you are negative because the negative allele is recessive. So if you were doing Punnet squares, by the way, you would do the Punnet square for rhesus completely separately. If you were doing monohybrid crosses, you would do the Punnet square for rhesus separately from the Punnet square for ABO. So, important to note. Okay. ABO is fun because you could have wacky stuff happen. Like if someone is type A, I'm going to clear this out because this slide is not ABO. But if you have one parent who is type A, that's their phenotype, and one parent who is type B, I might ask, could a parent who is type A produce an offspring with a parent who is type B and have that offspring be type O? And the answer would be maybe, right? Because people would assume if you have someone with type A and someone with type B and they have a kid, then it can't be type O. They have A protein and B protein. Maybe it would be type AB, who knows? But remember, right, you are probably thinking of this when I say type A, because remember, type A is the phenotype, not the genotype. And there are 2 different genotypes that can get you type A. And there are 2 different genotypes that can get you type B. And if you cross two parents, one heterozygous for A, one heterozygous for B, then it is fully possible to have a child that is type O. So, fun stuff with blood types. Okay, the last wrinkle I'll talk about in inheritance is real funky. And it has to do with color blindness. Now, red-green color blindness is a dominant recessive disorder. So, we'll use B for color blindness. The dominant allele is not red-green colorblind. And the recessive allele is red-green colorblind. And let's talk about how red-green colorblindness is inherited. So say you have one parent who shows the dominant phenotype of no color blindness. So not red-green colorblind. And one parent that is red-green blind. How will inheritance work? Right? Let's play the pedigree game. Okay, so we'll have a son that is colorblind. We'll have a daughter that is not, and a daughter that is not. And you will say, I've played this inheritance game before. We did this pedigree just a little bit ago. Look, let's work it backwards, right? The only way to be red-green colorblind is if you have two red-green colorblindness alleles. So here's the son, bam. Here's the female parent, bam. And the son got one allele from mom and got one allele from dad, right here. And there we go. Dad, in order to be not colorblind, has to be heterozygous, and all the daughters are heterozygous. Okay, so, you're a dummy. This was stupid. Why did you say this is another wrinkle? I just figured this out in record time. And bad news, hombre. That doesn't work at all. That doesn't, no. Here's how I'm going to mess it up for you. Because when you have one parent, if the male parent does not have color blindness, right? The male parent is not colorblind, and the female parent is colorblind. No matter how many offspring this parent combination has, no matter how many, if they could have 100 offspring, all sons, all of them, all 100% of sons are red-green colorblind. And 100% of daughters all of them, every daughter is not red-green colorblind. That's just how it works. That's it. That's all it is, folks. When the male parent is not colorblind and the female parent is colorblind, all sons are red-green colorblind. All daughters are not. What? What? How? No. No, that can't be right. Why is it that no matter how many times all the sons are red-green colorblind and all the daughters see red, blue, and green just fine? Why? Well, I keep emphasizing it here. There's another thing at play. Sons and daughters. That means we're talking about a completely different set of chromosomes. X and Y chromosomes, in which the X chromosomes code for being genetically female, and the Y chromosomes code for being genetically male. That is it, and that is pretty easy. So an XX codes for female, and an XY codes for male. You may ask, where are the YY? Uh, that is an inviable combination. That doesn't exist, right? In part because you need a male and a female, a sperm and an egg to produce a viable egg, so you can't combine two sperm to get a YY. But even if you could, um, the X chromosome carries genetic information that you need to live. The Y chromosome carries genetic information for a cock and balls and sperm. The X chromosome is actually necessary for being alive. So, you can actually be alive if you have a single X chromosome, but if you have a single Y chromosome, that fetus ain't developing. So, that's lethal. Anyway, that I digress. We are now dealing with XX and XY chromosomes in addition to the color blindness alleles. How on earth does this work? Well, I'm glad you asked. This is what we call a sex-linked trait. A sex-linked trait. What does it mean to be sex-linked? That means that the alleles for this disorder are literally carried on the sex chromosomes. So if all sons are red-green colorblind and all daughters are not, where is the color blindness allele carried? Which sex chromosome carries the color blindness allele? And I get a lot of people who instinctively say, well, if the sons are red-green colorblind, if all sons are red-green colorblind, then it's carried on the allele that determines whether or not you're male, the Y. But remember, the Y chromosome doesn't do much of anything, right? So the red-green colorblindness is an X-linked disorder. An X-linked disorder. That means I cannot just write out B's for color blindness. What I have to do is this: X, B, or X, B. And now this is going to make a lot more sense. Because remember, we had a father who is not colorblind. So they are carrying the dominant allele, and their other allele is Y, because they're male. So X, Y, and the dominant allele. And then you are connected with the colorblind female. So she is carrying two X chromosomes, and each one has the recessive allele. Now we see why all sons are colorblind, because remember, a male has to have an X or a Y, or an X and a Y chromosome. And where does the male get its Y chromosome? From the father. The father donates a Y chromosome. The Y chromosome carries no information about red-green vision. So the X chromosome in that male has to come from the female, and because the female is colorblind, all her X chromosomes carry color blindness. So if you are a male from that combination, that is your only possible genotype. A colorblind allele and a Y. And all the females get one X from mom that carries color blindness, and one X from dad that is the dominant allele. So all the females there we go. That is how females work. So, not bad. Uh, Pretty cool once you think about it. Uh, so, uh, this is pretty sweet. Uh, and this fully explains why all daughters have full red, blue, green vision. Also, what's noted is that all daughters are carriers. Carriers of color blindness. So that's kind of cool. So for these daughters, um, any sons that are born by these daughters will have a 50-50 chance of being colorblind. So if a female is a carrier for color blindness, um, then the offspring will have a 50-50 chance of being colorblind, right? So we can Punnet that out real quick. Really? Come on. There we go. Let's Punnet that out. Here's the female carrier. Here's the male. And we'll go ahead and not make the male colorblind. And we'll just Punnet this bad boy. So a female egg could either carry the not colorblind or colorblind allele, and the male sperm will carry the not colorblind allele and the Y. And thus, if it's sons, right, we have to have a Y chromosome. So when it comes to the Y chromosome, you will either get the X chromosome that does not have color blindness, or the X chromosome that does. And boom. 50-50 whether or not male offspring will be colorblind. Kind of cool. If the father's colorblind, then it becomes 50-50 whether or not female offspring are colorblind. So, all right. Pretty sweet stuff. Hemophilia, a disorder where you have problems clotting wounds, is also X-linked. In hemophilia, you do not produce, the hemophilia gene is a defective gene in which the allele does not code for clotting, essentially clotting factors. Gets more complicated than that, but do not carry genes for clotting factors. In other words, your blood don't scab, which is bad because if you get a cut and you can't scab up, you keep bleeding. Not a good thing. So, um, this is X-linked, so pretty scary stuff. what's interesting is that this is also, it appears to be incomplete dominant to a certain degree. Uh, uh, well, actually, no, not incomplete dominance. It has special rules. I don't actually go into this in this class, but um, you know how the X chromosome carries genetic information that is absolutely vital to being alive? Well, you only need one of those active. So during development, during fetal development, right, if you have two X chromosomes, uh, for a little while, both X chromosomes are active, and you're dividing and you're dividing and becoming a larger and larger clump of cells. And at a certain point, cells start to deactivate one of the X chromosomes, which is kind of cool. So if you're a female and you carry hemophilia genes, right? So here's a hemophilia gene, and you also carry non-hemophilia genes. Um, in your bones, in your bone cells, some of your bones will actually have the hemophilia carrying gene X chromosome deactivated, and some of your bone cells will have the hemophilia gene active and the normal one deactivated, because there's a 50-50 whichever one gets deactivated. And so females can actually have varying levels of hemophilia. It can go mild to extreme in females. And that's simply due 100% to how much of their bone marrow has the non-cell activated or the dominant allele activated versus the recessive allele activated. So heterozygote females can have mild to extreme. Homozygous recessive females all have extreme hemophilia. And males with hemophilia all have extreme hemophilia. So heterozygote females can have mild hemophilia if they win the lottery and most of their bone cells come from lines where the hemophilia allele was deactivated. So anyway, that's a lot of excess information, but it makes hemophilia particularly cool. Hemophilia was actually for a while called the royal disease. And that's because political alliances in like the medieval times through the Renaissance and whatnot were often made by marrying children from various royal and noble families out to other various royal and noble families. And Queen Victoria's lineage, Queen Victoria carried the gene for hemophilia. She was, and no, wait, was it her husband? Anyway, someone in the Victoria lineage had hemophilia, and then the kids started getting married out. And so suddenly, a lot of different royal families throughout Europe and Russia started showing multiple kids with hemophilia. And so it sort of became the royal disease. Anyway, your blood clots slowly, or if it's extreme, your blood straight don't clot. And if you get a paper cut, you're going to slowly bleed to death without like modern medical intervention. Good news, um, we can use bacteria, given the gene for the clotting factors, to mass produce clotting factors and transfuse clotting factors into people with hemophilia. So with modern healthcare, hemophilia is perfectly treatable and your blood will clot as normal as long as you go to the hospital every so often and get a transfusion of those clotting factors. Okay, what are the Y-linked disorders, you ask? Not a lot. Like I said, the Y chromosome doesn't do much, right? It governs uh, biological male development. If you have the right gene that gets activated at the right time, there's a pulse of testosterone during fetal development, and you get biological male development. That's what the Y chromosome do. There is, however, one Y-linked recessive disorder that I could find that does not result in infertility. There are Y disorders, but there's not much on the Y chromosome, and if you have a disorder of that gene that causes you to develop as male, generally it also causes you to develop as infertile. But however, we have here hypertrichosis of the pinna, and that is a Y-linked recessive disorder that does not cause infertility. Hyper, lots, trick is often the prefix we use when referring to hair or hair-like stuff. Pinna is your outer ear. So hypertrichosis of the pinna is excessively hairy ears. Why are there so few? Because again, the Y chromosome don't do much. But it is funny. If you have the Y-linked disorder, right? So hypertrichosis of the pinna, we'll call it trichosis, a T. So this is again recessive. So not hairy ears, not excessively hairy, and excessively hairy. And if you are a male, there you go. You could be XY with the excessive hairy ear allele, or and here's XY with the excessively hairy ear allele, and here's without. And if you've got that excessively hairy ear allele, congratulations. Every male in your lineage, every male directly from your Y chromosome, will always have excessively hairy ears. Yeah, your son can marry a woman without excessively hairy ears, and his sons will have excessively hairy ears, and they can marry women, they can have offspring with females that do not have excessively hairy ears and whatnot. So, it doesn't matter who they choose to have offspring with if they're passing on their Y chromosome. That Y chromosome has excessively hairy ears. Anyway, the SRY gene is that gene that triggers that testosterone pulse that results in development as a biological male.